Alright, so we're going to pick up now with our next section on genetics. And starting off this section is going to be a quick overview of a concept called pleiotropy. So pleiotropy, like I mentioned at the end of the previous section, is kind of the opposite of polygenic inheritance. So if you think back to polygenic inheritance, we had a situation where many genes controlled the outcome of a single phenotypic trait. So 3 genes, for example, have the effect on determining how much melanin is produced and therefore the tone of the skin in somebody who produces melanin, which is, of course, almost everybody. With pleiotropy, we actually have now a single gene that has multiple effects. So now, depending on which allele you carry, this one particular gene might have an influence over 20 or 30 different features of your phenotype, which might seem a little bit outlandish, but it actually is something which can happen. So, for example, one of the best examples of pleiotropy is the SRY gene that human males have. So the SRY gene is a gene that really, really early on in gestation or development or pregnancy, if you have an SRY gene, and if you do, you're going to be male, that SRY gene is activated, and that triggers a whole lot of changes to take place to convert you from being female into being male. So that's a pretty wide range of phenotypic, anatomical, hormonal, physiological, behavioral traits that all stem from the presence of a single allele, that being the SRY gene. Now, if you don't have SRY and you're female, of course you have a whole different set of traits, right? So in that case, this is actually a really good example of pleiotropy. A couple of other examples of pleiotropy involve conditions or diseases. One of them is cystic fibrosis, which if you have 2 recessive copies of the allele for cystic fibrosis, there is a wide range of phenotypic traits that makes it hard to breathe and makes you very prone to infection. Essentially what happens is your mucus-producing cells in your respiratory tract produce a mucus that's way too thick, and therefore you're unable to get it out of your respiratory tract like you're supposed to, and whatever pathogens might be trapped in that mucus can increase the risk of infection. So if you produce normal mucus, it works great. It traps the pathogens, the bacteria, the viruses, the spores, whatever you breathe in, and then you kind of cough, or even by itself, it moves the mucus up and out. Your respiratory tract moves mucus up and out, and you're fine. But if you produce a very, very thick mucus, then not only is breathing and gas exchange difficult, but you're much more susceptible to infection as well. Now, kind of interestingly, if you only have one allele for cystic fibrosis, you actually end up having the symptoms, but at a much, much, much lesser level. So you're able to actually function having, excuse me, one copy of that cystic fibrosis allele. The other thing that's interesting about this is if you have a single copy of the cystic fibrosis allele, you actually are more resistant to a different disease called tuberculosis. And so tuberculosis is caused by a bacterium called Mycobacterium tuberculosis that has a really difficult time living in the lungs of a person who has low-level cystic fibrosis. So living with one normal copy of this gene and one cystic fibrosis version of the gene makes you resistant to tuberculosis, and it also gives you low-level cystic fibrosis symptoms, which are fairly easily dealt with if you have some reasonably decent medical care. Sickle cell anemia is an even more famous example where there's kind of like 3 options here, right? So you can inherit 2 copies of the normal allele for hemoglobin, in which case you have normal hemoglobin and absolutely no symptoms or signs of sickle cell anemia. If you have one normal and one sickle cell allele, then you have kind of like mild sickle cell anemia with symptoms, but again, the symptoms are fairly survivable. And if you have 2 copies of sickle cell allele, you're going to die of sickle cell anemia. There's really no way around that. Even if you have medical care, 2 copies of sickle cell is very, very difficult to deal with if you have it. So the other thing that's interesting with this one, kind of like we saw with cystic fibrosis, if you have a single copy of the sickle cell anemia allele, it also makes you resistant to malaria, which is a really, really big killer of people. Something like 400,000 people a year, give or take, die of malaria. But if you have a single copy of the sickle cell allele, you actually have some partial protection against malaria. So we'll get to that kind of description of that scenario a little bit later on when we cover chapter 20. So with sickle cell, and again, we have red blood cells, which these 3 cells here look fairly normal, but we have one cell here, which is a sickle cell, and another one up here. And the reason for these cells having this shape is that the hemoglobin they carry is abnormal, and therefore it causes a distortion of the cell itself. So with those sickle cells in circulation, we have a shorter lifespan of red blood cells, we have clogged blood vessels, especially the very, very tiny capillaries in the body, We also have an overworked spleen because the spleen has to recycle the dead red blood cells a lot quicker because they're not living as long. Those symptoms lead to these symptoms, and those symptoms lead to these symptoms. And so, again, what is pleiotropy? It is a single allele that causes a huge range of effects on the phenotype, as we're seeing here. This is because of one allele. We see all of these different symptoms showing up. The one that's not shown up on this slide is death. So it's a very wide range of effects on the phenotype just because of the presence of this one sickle cell allele. Now, another scenario, which we haven't talked about yet, is what happens if you have more than just two alleles in the population? What happens if you don't just have two? You've got maybe a dominant and a recessive, and that's all there is. What happens if you have 3 or 4 or 5 or 10 different alleles in the population? That's going to make it a bit more difficult to track the genetics of the inheritance in this population. So a really good example of this one is the ABO blood type system in humans. So first thing here, you probably have heard of A positive or A negative or B positive or B negative. We're not worried about the positive and negatives in this scenario. That's actually a very different gene. And so what they do is they combine the ABO property of the blood with the Rh factor property of the blood to give you an indication of whether you're AB positive or AB negative or O positive or O negative or whatever. We're not worried about positives and negatives. We're just worried about the A's, B's, and O's in the population. So clearly then, we can't have a simple dominant recessive relationship because we have 3 different alleles. And so even though each person inherits one allele from each parent, this creates a slightly more complicated scenario when it comes to the inheritance of this particular trait. So this not only is a really good example of multiple alleles in the population, but also secondarily an example of something called codominance, where the A and B alleles are both dominant over the O allele, and they're codominant with each other, which means if you have an A allele and a B allele in an individual, they actually express both of those alleles, as opposed to dominant recessive, where the dominant gets expressed, the recessive doesn't. With A and B together, they're codominant, so they both get to show up as part of the phenotype, and O is recessive. So to have O-type blood, you have to have 2 copies of the O allele. Otherwise, if you have anything else, you're not going to have type O blood. So the way this works, we've talked about chromosome 9 before, and the presence of the blood type gene on chromosome 9, and this is the one that we're talking about in this scenario. In terms of the different genotypes you can have as an individual, if we kind of look at this table here, so this genotype means you inherited A alleles from both parents. This genotype means you got an A allele from one parent and an O allele, little I is O allele, from the other parent. So in both cases, whether you're homozygous for A or heterozygous for A and O, you're going to have type A blood. And we'll talk about what this is here in a second. If you're type B blood, you're either homozygous for B or you're heterozygous for B and O. So in both cases here, you would have type B blood, and you'd have galactose as the carbohydrate on the outside of those cells. If you have A and B together, so if you got an A from one parent and a B from the other parent, you have A and B together, you are type AB blood, and you have both of the carbohydrates that are listed above. And if you got an O allele from both parents, then you have type O blood, in which case you don't have any carbohydrates of this type on the outside of your blood cells. So the difference between galactosamine and galactose, and galactose, and just nothing there on the outside of the cells makes a really big difference when we're talking about transfusions and who can give blood to whom and why that is. So if you're a type A person, type A blood, you can get blood from another type A donor or you can get blood from a type O donor. But if you have any blood that has the B galactose carbohydrate on it, what's going to happen is your immune system is going to reject those cells that are expressing that B on the surface, and that's going to cause what's called an agglutination reaction, where the cells clump together and they're destroyed, and they can cause clots, and clots then lead to strokes and heart attacks and embolism, and all kinds of really bad stuff starts to happen when you have the wrong blood type introduced into the system. Similarly, if you're type B blood, you can get blood from a type B person or a type O person, because the thing with type O, we call O the universal donor because it doesn't have any carbohydrates on its surface in the red blood cells, so there's nothing for the immune system to recognize and attack. So type O blood can go to anybody because those cells don't express anything on their surface when it comes to this particular carbohydrate. If you're type AB blood, in a sense, you're kind of lucky because you can get blood from anybody. So if you have type AB, your body's going to recognize the A's. If you get a donation from a type A person, your body would recognize the B's. If you get blood from a type B person, AB, of course, is fine as well. And O is universal donor, so you could also get that if you're type AB. So that's a little bit of a side note on what blood types actually are. But it's a really good example of both codominance and a situation where we have more than two alleles in a population. And so this slide is again just kind of showing us what the kind of a schematic or cartoon view of, type A blood. You've got this blood cell with a bunch of A markers, galactosamine markers on it. If you're type B blood, you've got a bunch of galactose markers on it. If you're AB, you've got both galactosamine and galactose on the surface. And if you're type O, you don't have any of those glycoproteins. And again, that makes a really big difference when it comes to who can donate blood to whom and who can receive blood to whom or from whom. So again, it's a really good example of a lot of concepts here in biology. So this is kind of a similar, talking about codominance, where when A and B are together, neither one is dominant, and they both get to show up as part of the phenotype when you have a type AB blood person. All right, so that was codominance and multiple alleles. Another example, which is kind of similar to that, is incomplete dominance, where, again, it's a situation where you have two alleles that both get to show up in the phenotype because one of them is incompletely dominant over the other one. All right, so what you eventually end up with here is a bit of almost like a hybrid phenotype where it's got some properties of both alleles present in the phenotype or the physical appearance. So in some plants, we talked about garden peas before with Mendel and his stuff. Pea plants had purple and white flowers, and the purple allele was dominant over the white allele, and it was a very clear-cut scenario. Well, in other flowers, like carnations and snapdragons and a few other things, hibiscus is another one, if you have a plant that has a red allele and a white allele present, it's going to produce pink-colored flowers. And pink is kind of a combination or a mixing of red and white to produce pink. All right, so we'll take a look at a scenario here and kind of work through this with these hibiscus flowers here. So to have a red flower, hibiscus has to have two red alleles from its parent. And to have a white flower, hibiscus has to have two white alleles from each parent. If you get a white and a red together, that gives you a pink flower plant. And if you cross two pink flower plants together, you actually get one red, two pink, and one white. So you get a 1 to 2 to 1 ratio of red to pink to white in the offspring. So again, like the Punnett square that we looked at before, if this parent, say this is the male parent here, if the sperm cell carries the R allele, the red allele, and the other parent also, the egg cell carries the R allele, then you're going to get both reds together. That's going to produce homozygous red, which gives you red flowered plants. If this one gives up red and this one gives up white, then you're going to get a pink flowered offspring. Same thing here. And if both parents give up white alleles that show up in the phenotype, then we're going to have homozygous for white, and that plant would have a white phenotype as well. So that's a good example of incomplete dominance, which sometimes muddies the waters a little bit when you're looking at how these traits are inherited and how they're expressed as part of the phenotype. Now, something else to kind of think about here, which is also very important, is the effect of the environment. And so the environment can produce effects in a couple of ways. I mean, sometimes the environment itself can have effect on the phenotype. So, for example, if you were to dye your hair blue, that would be a phenotypic change. That would be part of your phenotype, having blue hair. But that effect of your environment would not be passed on to your offspring. So in that case, it's an environmental influence that's not involved in any sort of genetic capacity at all. In other cases, you can actually have the environment affecting how the different alleles are expressed. So in this scenario on this slide, we have Himalayan rabbits and also Siamese cats, like in the picture here. And so the gene for an enzyme called tyrosinase is activated preferentially in parts of the body that are below a certain threshold temperature. So for example, the tips of the ears, the tip of the nose, the tip of the tail, those are the parts of the body in these animals that tend to be the coolest because they're farthest away from the core. Now, the cells that are in those parts of the body that are expressing tyrosinase express more of it because those parts of the body are colder. And for that reason, the skin and the fur in those parts of the body takes on a darker appearance. Right? So somewhere between about 30 and 33 degrees Celsius, there is a threshold where if you're below it, the enzyme's activated. If you're above it, the enzyme is inactivated. You don't get pigment. And so you end up with both these cats and rabbits having this characteristic appearance of white over most of the body, but kind of a blackish or dark brownish fur at the extremities. So in this case, the environment's influencing gene expression, but in other cases, of course, the environment can affect the phenotype directly. So, it's not all about genetics. It is, a lot of this is also due to the environment. There's been a long debate about nature versus nurture in a lot of different types of disciplines, psychology versus biology and those kinds of things. And it's really hard sometimes to separate out nature from nurture or genetics versus the environment, because to do that, you have to have a control, right? So The ideal scenario would be to take a pair of identical twins who genetically are the same, right? So they're genetically homogeneous. They both have the exact same sets of alleles, but you would raise them in different conditions and then come back maybe after 20 or 30 or 40 years and see in what ways they're similar and maybe what ways they're different. Similarities then would be the result of genetics because they're raised in different environments, in different homes. Any differences presumably then would be due to the environment of being in different homes. Now, of course, we can't do that. We're not allowed to take babies from the hospital and separate them at birth and raise them in different conditions. That's a horrible, horrible idea in reality. But there actually have been cases where this has occurred for other reasons, not because of any sort of scientific investigation or anything like that, but due to other kind of social issues or personal issues. Twins separated at birth can be reunited maybe a few decades later, and what's really kind of bizarre is not only do the people end up looking similar and behaving similar, sometimes they even have similar professions. What's even more bizarre is sometimes these separated at birth twins can actually have similar looking spouses, which is really kind of crazy to think that your choice of mate is at least somewhat genetically determined. Kind of crazy to think about it that way, but that does appear to be at least partially the case. Now, earlier on in one of the previous sections, we talked about sex-linked traits. It turns out that in mammals and birds and some of the insects, we have sex chromosomes that determine the sex of the offspring. All right, so when I say sex here, we're talking about the biological sex. We're not talking gender, which is a much more of a continuum sort of a thing where, there's other kinds of things that influence the gender of a person versus the sex of the person. The sex of the person is clear-cut at birth, either male or female. Any sort of differences beyond that are outside the realm of what we're talking about in this slide. Okay, so just right off the bat, I want to make that disclaimer. So in mammals, in our case, if you are heterozygous for the sex chromosome, so in other words, if you have an X and a Y, you are going to be biologically male at birth. If you are homozygous for the sex chromosome, so in other words, if you have two X's together for the sex chromosome, then you are going to be genetically, biologically female at birth. So it turns out that even though these two chromosomes, like in this karyotype picture here, even though these chromosomes behave as if they're homologous, they're not actually homologous chromosomes. They don't actually share all that many genes. So remember what the definition of homologous is. It's two chromosomes that have the same genes, even though the versions of the genes can be different. The X and Y chromosome really don't have that many genes in common. I mean, look at the size of these two chromosomes. The X chromosome is pretty big. It's got something like 1,000 genes on it. The Y chromosome is tiny. It's actually the smallest chromosome that humans have ever. It's made up of like 78 genes. So it's a very, very small chromosome. But even though they're not truly homologous, they do behave as if they were homologs during meiosis. So during meiosis, when you have the homologs lining up on the metaphase plate, during metaphase 1, for example, it's the X and Y chromosomes that are lining up together on the metaphase plate. So it's kind of interesting there to see that they're not really homologous, but sometimes they actually behave as if they are. But what's more important is that these sex chromosomes, especially for males, if you have an X and Y situation, if you're a biological male, then because the X chromosome has like 1,000 genes on it, the Y chromosome only has 78 genes on it, clearly there's going to be a bunch of genes on the X chromosome for which you only have one copy. Right? You've only got one X chromosome, so whatever mom gave you on that X chromosome, that is what's going to be expressed. Whether it's dominant or recessive, it doesn't matter because there is no homologous gene on the Y chromosome. Whatever you have on that chromosome is what you're stuck with for males. Now, if you're female, if you have two X chromosomes, if you inherit, say, a negative recessive allele on one part of an X chromosome that you got from one parent, there's at least a decent shot that the other parent might have given you the normal version of the X chromosome, in which case you would be phenotypically normal. Males don't have that luxury because we only have one X chromosome, so whatever we got from our mom in terms of genes on the X chromosome is what's going to be expressed as part of the phenotype. All right, so again, these chromosomes, even though they act like it, are not really technically homologous. All the other chromosomes in the genome are referred to as autosomes, so you'll hear me use that term now. We've introduced that term. Autosomes are the non-sex chromosomes. All of the chromosomes except for the X and the Y. So in reality, that means chromosome number 1 all the way through chromosome 22 are the autosomes, and that last pair of chromosomes are the sex chromosomes. Now, when it comes to sex determination, kind of interesting idea here is who actually determines the sex of the offspring? Is it dad or mom that does it? Well, we can kind of answer that question by looking at this slide here. So mom, in her, what we're going to call egg cells, they're not really egg cells, we're going to call them that. In her egg cells, she only has X chromosomes because that's all she has in her autosomes. So the only thing she has to give to the offspring are X chromosomes. Dad, on the other hand, has an X chromosome, which he got from mom, and a Y chromosome that he got from dad. So half of dad's sperm cells carry an X, the other half of dad's sperm cells carry a Y, which means at birth, the sex ratio of humans, if you look at a big enough sample size, should be about 50-50 because on average, half of the sperm cells carry an X, half carry a Y, which means we get half females and half males on average at birth. Now, does it always have to be exactly 50-50? Absolutely not. If you have a very small sample size, especially if you have, if you look at 10 pregnancies at random in a population, there's a chance they might all be male or might all be female. But if you look at 10,000 or 10 million births in a population, then there's a really good chance it's going to be really, really close to 50-50. It's usually not exactly 50-50. There's a few aspects of male infant survival that are, and actually embryonic and fetal survival that make males a little bit less likely to survive and be born than females, but it's actually really close. It's like 49% males and 51% females or something like that. Really close to being 50-50. So let's talk about those sex-linked genes. All right, that X chromosome is big, the Y chromosome is small. And so again, whatever genes you have on the X chromosome, if you're male, that is what's expressed in the phenotype. Now for females, you have two X chromosomes, you're back to simple dominant recessive relationship where, depending on what's on the other chromosome, that will determine which of the two genes are expressed in the phenotype of an individual. The Y chromosome only has 78 genes, so it's a much smaller, much more compact chromosome than the X chromosome is. All right, so again, females are the homozygous sex, they are XX, males are XY, and that's kind of how we determine our sex. Now, in terms of sex-linked diseases, again, these occur far more frequently in males than females, and one really good example for this is color blindness. You guys are hearing my dogs talk a little bit here as well. Color blindness is almost always seen in males. All right, so again, the gene for color blindness, as you might sort of think of it, is on the X chromosome. If a male inherits the colorblind allele from his mother, he is going to be colorblind. There is no equivalent on the Y chromosome that can bail out a male that inherits the colorblind allele from his mother. On the female side of the equation, on the other hand, well, since females have two X chromosomes, even if they got a colorblind allele from one parent, there's a really good chance the other parent gave them the non-colorblind allele, and for that reason, then they're not going to be colorblind. So females tend to be carriers. Females tend to pass the trait on to their male offspring or their sons, but females themselves typically do not actually express the effects of these sex-linked traits. Hemophilia is another really good example. So hemophilia involves a situation where different proteins in the blood are not produced the way they're supposed to, and for that reason the blood doesn't clot, and it can actually be very, very problematic for people who are hemophilic because any sort of cut or bump or bruise or scrape will just continue to bleed, and if you don't have some sort of outside clotting factor added to the wound, the blood will just keep leaking, and you're going to eventually bleed out, even with a fairly minor wound, never mind a very large wound. So hemophilia is a disease that's been seen in royal families for many, many hundreds of years because royal families tend to not marry outside the family and not produce children outside the family so much. So that is another can of worms we could open up, I guess, at another point in time as far as why inbreeding is not such a good thing. And there's the ickiness factor of it, first off, but there's also some really good genetic and natural selection reasons why individuals should breed outside the family because of what are essentially harmful recessive alleles. If you limit your choice of mates to your close relatives, then any sort of recessive, especially harmful recessive alleles that show up in the population might have a greater chance of showing up in the offspring, and so that could lead to some children that have some negative symptoms in a way. All right, so in this particular case, this is actually a very famous situation where we've got Queen Victoria of England, who was a carrier for hemophilia. So she is essentially, she got one normal allele from one parent and one colorblind allele from the other parent. She herself was not colorblind, but she ended up passing it on to one son who was fully colorblind and two daughters who, even though they weren't colorblind, they were carriers. And again, they were able to pass it on to their offspring. The sons who got the colorblind allele ended up being colorblind. Some of the sons were perfectly normal. And of course, daughters can be carriers. Now, another question you might ask is, well, is it even possible to have a female who has hemophilia or colorblindness? And it absolutely is possible, but it's not probable. It's very uncommon to have a female wind up with both harmful recessive versions of an allele and actually express a phenotype like hemophilia or like color blindness. I'm not sure what happened there, but okay. So that tends to be fairly uncommon when we have these sex-linked traits where you actually have females that express the harmful condition. Now, something else we're going to talk about that kind of goes back a little bit to Chapter 11 is what happens when you have a mistake during meiosis. We talked about non-disjunction. I said non-disjunction is a situation where something doesn't separate the way it's supposed to during meiosis. So what we have on this slide are three different scenarios. We have a normal type of meiosis, which has, this particular cell has 4 chromosomes. So for this species, whatever it is, it's not human, it's only got 4 chromosomes. It's going to go through meiosis 1. And it's going to produce 2 daughter cells that are now haploid, but still have sister chromatids attached. And eventually both of these 2 cells go through meiosis 2, and they produce daughter cells which are not only haploid, but only have a single chromosome, or a single chromatid for each of those 2 chromosomes. So we have 4 haploid cells that all have exactly 2 chromosomes, just like they're supposed to have, because the diploid condition is 4, the haploid condition should be 2. Those cells work out like they're supposed to. Now in the second scenario, we start off the same. This diploid cell has 4 chromosomes, but look what happens in meiosis 1. These 2 homologs here line up together and they don't separate. So these 2 chromosome 1s are going to stay together on the left side of the metaphase plate. So when this cell divides, this daughter cell gets 2 big chromosomes, 2 chromosome number 1s, and 1 chromosome number 2. So it actually has 3 chromosomes, and the other daughter cell only has 1 chromosome. So what we actually end up producing here, eventually when we get through this, is gametes that have the wrong number of chromosomes. And if you think back in Chapter 11, we had a name for that. It was described as being aneuploid, where cells were produced with abnormal numbers of chromosomes. A chromosome that has an extra chromosome, or a cell with an extra chromosome, or a cell that's missing a chromosome. It's that kind of situation where it's not having an entire extra set of chromosomes, it's just having an extra chromosome, or maybe missing a chromosome, for example. If any of these cells were involved in a fertilization event as gametes, the zygote that was produced very likely would not survive and it would die almost immediately because, again, having the wrong number of chromosomes is not a recipe for success for any species. Now, if the mistake that occurs is during meiosis 2, we'll take a look at that scenario here. We start off with the same. We have a cell with 4 chromosomes. We produce a daughter cell that's going to divide and produce 2 daughter cells that each have 2 chromosomes. So far, so good. These 2 cells over here, in the normal case, look just like these 2 cells over here, where we're going to see non-disjunction happening because non-disjunction hasn't happened yet. When these 2 cells divide, what happens next is these 2 chromatids don't line up on opposite sides of the metaphase plate like they're supposed to. They both go left. This one goes left, this one goes right, and we end up with these 2 daughter cells being aneuploid. This one has an extra chromosome, this one's missing a chromosome. The other one, because the chromosomes lined up correctly on the metaphase plate, it's perfectly fine. And so these 2 gametes are going to be perfectly normal. So this situation is actually not quite as bad because you do actually get some normal gametes produced during this round of cell division. So another question to think about before we kind of wrap up this section is, well, what does this mean for someone's overall fertility? How often does this actually happen? And if it happens, does that mean they're sterile? Does it mean they're less fertile? Well, this is only a single cell doing this, right? And so for males in particular, males produce hundreds of bazillions of gazillions of sperm cells during their lifetime. Those are, metric measurements, of course. If this happens once or twice every now and then where you get a couple of aneuploid gametes, it's really not a big deal because, for example, in a single ejaculation, there's about 200 million sperm cells. And so if you have this non-disjunction thing happening once, or even this one happening once, okay, you've got 4 cells out of 200 trillion that are abnormal, or 2 cells out of 200 trillion that are not normal. I'm sorry, not trillion, billion cells that are not normal. It's 2 billion cells. It doesn't by any stretch of the imagination mean you're less fertile or sterile or anything like that. It just means you've got a couple of bad cells in there. And the chance of either of those 2 cells actually being the ones that fertilize the egg, chances are not very good that that's going to take place. So, unless this gets to be really, really common in gametogenesis, even for females who don't produce nearly as many gametes, it's really not going to be that consequential. One thing to think about and to note, though, is that as we get older, the chances of making these kinds of mistakes becomes greater. And so the older you are at the point where you produce a gamete that is involved in fertilization, the more likely you are to have these abnormal gametes produced and therefore end up with a zygote that has an irregular number of chromosomes and therefore is very unlikely to survive. So like I said, we're going to wrap up this section here, and I'll have one more short section for you here briefly. I'm not talking about that, though. And we'll finish out this chapter in just a second.